Today, we will tackle about the Human Genome Project. At a glance, the human genome, salient features of human genome. What was Human Genome Project? The milestone of Human Genome Project, the goals of Human Genome Project, and the issues of concern, and the future challenges of Human Genome Project. What was the Human Genome Project? The human genome is the complete set of the genetic information of humans, Homo sapiens. The human genome is by far the most complex and largest genome. It is the size spans a length of about 6 feet of DNA, containing more than 30,000 genes. The DNA material is organized into a haploid chromosomal set of 22 autosomes and one sex chromosomes, the X or Y. Salient features of human genome. Human genome consists the information of 24 chromosomes, 22 autosomes plus X chromosomes plus 1 Y chromosomes. In Homo sapien, 2N is equal to 2X equals 46. The human genome contains over 3 billion nucleotide pairs. The human genome is estimated to have about 30,000 genes. Average gene consists of 3,000 bases, but sizes of genes vary greatly, with the largest known human gene encoding dystrophin containing 2.5 million base pairs. Only about 3% of the genome encodes amino acid sequence of polypeptides, and rest of it junk, repetitive DNA. The functions are unknown for over 50% of the discovered genes. The repetitive sequences make up very large portion of human genome. Repetitive sequences have no direct coding function, but they shed light on the chromosome structure, dynamics, and evolution. Next, chromosome 1 has most genes, 2,968, and Y chromosomes has the lowest, got 231. And almost all nucleotide bases are exactly the same in all people. Genome sequences of different individuals differ for less than 0.2% of base pairs. Most of these differences occur in the form of single base differences in the sequences. These single base differences are called single nucleotide polymorphisms, or SNPs, and one SNP occurs at every 1,000 BP of human genome. About 85% of all differences in human DNAs are due to SNPs. This image shows the human chromosome 1 genetic map. It is hard to understand what was this because we're not an expert. What was Human Genome Project? The Human Genome Project was an international research effort to determine the sequence of the human genome and identify the genes that it contains. The U.S. Human Genome Project is a 13-year effort which is coordinated by the Department of Energy, or DOE, and National Institute of Health. The milestone over years of the Genome Project. The birth of the Human Genome Project was on 1986, and project initiated as joint effort of U.S. Department of Energy and the National Institute of Health was on 1990. And 1994 is the Genetic Privacy Act to regulate collection, analysis, storage, and use of DNA samples and genetic information is proposed. And Wellcome Trust joins the project was on 1996, and Celera Genomics is a private company founded by Craig Venter, formed to sequence much of the human genome in 3 years. In 1999, completion of the sequence of chromosomes 22, the first human chromosomes to be sequenced. And in 2000, completion of the working draft of the entire human genome. And 2001, analysis of the working draft are published, and HGP sequencing is completed and project is declared finished 2 years ahead of schedule was on 2003. If there are milestones over years of the Genome Project, of course, there are goals of the Human Genome Project. First is to identify all the genes in human DNA. Second, to develop a genetic lineage map of human genome. 3. To obtain a physical map of human genome. Number 4. To develop technology for the management of human genome information. And number 5. To know the function of genes. Number 6, determine the sequences of the 3 billion chemical base pairs that make up human DNA. And number 7, store this information in public databases. Number 8, develop tools for data analysis. And last, transfer related technologies to the private sectors. If there are milestones of years of the Human Genome Project and the goals, of course, there are issues of concern when it comes to ethical, legal, and social issues of the Human Genome Project. The first issues of the Human Genome Project is the fairness in the use of genetic information, privacy and confidentiality of genetic information, psychological impact, stigmatization and discrimination, and reproductive issues, clinical issues, uncertainties associated with gene tests for susceptibilities and complex conditions, and fairness in the access to advanced genomic technologies, conceptual and philosophical implications, health and environmental issues, commercialization of products, education standards and quality control, and last is the patent issues. And that is all the issues of concerns when it comes to ethical, legal, and social issues of the Human Genome Project. The Human Genome Project is still on process when it comes to research, so that Human Genome Project was still searching the things inside the DNA or the genes of a human being, because there are so many things that still don't know. The first future challenges was how many gene number, or what were or where is the exact locations and the functions. And number 2, gene regulation. Number 3, chromosomal structure and organization. And number 4, non-coding DNA types, the amount, the distribution, information, content, and functions. Number 5, coordination of gene expressions, protein synthesis, proteomes, and post-translational events. Number 6, predicted versus experimentally determined gene function. Number 7, evolutionary conservation among organisms. Number 8, disease susceptibility prediction based on gene sequence variation. Number 9, genes involved in complex traits and multi-gene diseases. And last, developmental genomics. That is all the future challenges that we still don't know. And that is all the Human Genome Project, and good morning and thank you for listening.